Hello, everyone. My name is Tyler Harmon, and I'm a master's student at Grand Valley State University. And I'm very excited to present my research here at this year's Global Coral Reef Week, and I look forward to all the other amazing presentations. Today, I'll be discussing part of my thesis research, which looks at seasonal thermal resilience in the temperate coral Astrangia poculata. Over the past several decades, anthropogenic means have been exacerbating climate change due to fossil fuels, deforestation, agriculture, and so forth. These practices have increased the amount of greenhouse gases, such as CO2, in the Earth's atmosphere, causing increases in solar radiation, subsequently warming our planet at accelerating rates. Many ecosystems and organisms have been exposed to these increased temperatures, causing negative consequences. In marine systems, tropical corals are at the forefront of these increased thermal regimes. One of the main outcomes of thermal stress on corals is a process known as bleaching. Corals have an obligated symbiotic relationship with algal cells, which supply corals with large amounts of their energy needs. When exposed to these warming temperatures, coral hosts expel their algal cells, leaving the coral a bleach white and under the threat of mortality. Many studies have pinned reactive oxygen species as the indicator of these bleaching mechanisms. These thermal stress events lead to the expulsion of algal symbionts, and the main reason is due to reactive oxygen species. These are a normal byproduct of photosynthetic functions in these algal cells, but can increase in concentration when exposed to large amounts of energy, either with light or with temperature. The increase in energies damage and disrupt photosystems 1 and 2 in the thylakoid membrane and the Calvin cycle, leading to increasing amounts of ROS in the form of singlet oxygen and hydrogen peroxide. Coral hosts then identify these impacted algal cells and expel them to prevent further damage to themselves, as these ROS components can leak into the host tissue, damaging the mitochondria and resulting in host cell apoptosis. These bleaching and thermal stress studies have been centered around tropical coral species due to the urgent need of saving these coral from extinction. However, reefs exist in other climatic regions of the planet and could be susceptible to these impacts in the future if climate change continues its business as usual concept. This leads into our study species known as the northern star coral, or Astrangia poculata. Thermal resilience is not as well known in temperate corals, although studies have been gaining popularity in temperate species over the past decade. This species has been identified as a model organism due to its facultative symbiotic properties, meaning it can live with and without its algal symbiont, in addition to living in different thermal extremes. This species' northern range occurs in Cape Cod, Massachusetts, and occurs as far south as southeastern Florida and the northern shores of the Gulf of Mexico. So why study temperature resilience in temperate species? Well, many studies have identified increases in thermal stress events within areas of known temperate species, identifying the need to further the research. In addition, many cellular processes have not been researched within temperate species, and this can help identify the influence of symbiosis and potentially apply to other areas of thermal stress research in corals. Our overall goals are to identify how ROS changes between thermal stress events and between symbiotic states, as well as between seasons. Most of the ROS research has been focused on obligate symbiotic coral. So how will this facultatively symbiotic coral influence our ROS production? It's also been shown in Sharp et al. 2017 that seasonal changes occur specifically within the microbiome of the species. So how will season impact ROS production? In addition, we will compare photosynthetic efficiency and symbiont density data to back up our findings. Our main hypotheses are thermal stress treatments will increase these ROS concentrations compared to the ambient temperature treatments, as well as symbiotic colonies of Astrangia will be more impacted by thermal stress due to ROS concentrations, resulting in decreased photosynthetic efficiency and symbiont density. Our collections took place in Narragansett Bay in Rhode Island, specifically at Fort Wetherill State Park. We made collections in July of 2019 and February of 2020. This video shows the eutrophic, rocky substrate environment that these coral call home. You can see mostly aposymbiotic colonies shown here from this video taken several months ago in February. Once collections were complete, we brought them back to our custom recirculating aquarium system at the Annis Water Resource Institute here in Michigan. After weeks of acclimation to the system and the fragmentation process, we distributed these fragments randomly and evenly as possible between two different temperature treatments. One at 18 degrees Celsius, which is their mean temperature found at the Rhode Island location, and one at 26 degrees Celsius, which simulates future climate stress, about plus 2 degrees Celsius from a business as usual scenario. These were exposed over a 10-day period to detect changes over time. The few measurements we conducted was firstly measuring photosynthetic efficiency of these coral, or its maximum quantum yield, to identify the overall health of its photosynthetic processes in photosystem 2. Our second form of measurements was the photoquantification technique. This is a non-invasive method that was developed by Winters et al. In 2009, which uses photographic methods to estimate symbiont density in the form of pixel intensities. A custom MATLAB script was used to make photographic corrections, and then selecting areas on the coral tissue to generate data, as you can see off to the right. Thirdly, we used imaging flow cytometry to identify changes in ROS concentrations by using an Amnis image stream flow cytometer and a general oxidative dye, CmH2DCFDA. Here you can see individual pictures taken of items like algal cells and host tissue up in the top right, as well as specific channels showing fluorescence. Channel 2 in the middle shows Fitzy fluorescence, as well as channel 5 off to the right indicates autofluorescence. This gave us the ability to set gates within our sample collections to easily identify differences between control, no dye, and stained samples, which you can see compared in the bottom right. Now for the best part, which is our results. The overall analyses with maximum quantum yield did not show any significant differences between thermal treatments, but we did find higher rates in summer compared to winter symbiotic colonies. However, no differences were found between season with these aposymbiotic fragments. Overall, symbiotic fragments had higher maximum quantum yield rates compared to aposymbiotic fragments. Linear trend lines show that thermal treatments with aposymbiotic fragments increase the overall maximum quantum yield rates over time, in addition to ambient temperature treatments from the winter aposymbiotic fragments. We did not find any linear trends within our symbiotic fragments, regardless of temperature treatment and season. Our symbiont density and pixel intensity data also did not show significant differences between treatments in the summer collections of our symbiotic fragments, and no differences for all of our aposymbiotic fragments, regardless of season. We did notice a significant difference between our symbiotic fragments from the winter collections, showing that these thermal treatments actually benefited symbiont density compared to ambient temperature treatments. Overall, symbiotic fragments had much lower pixel intensities compared to aposymbiotic fragments. No changes were identified in our summer aposymbiotic fragments, but we did find slight inverse trends in our summer symbiotic fragments, where the thermal treatments increased in pixel intensity over time. We did see similar and more pronounced trends in our winter symbiotic fragments, and slight decreases in our winter aposymbiotic fragments, implying the influence of higher temperatures on our winter coral collections. Our aposymbiotic ROS fluorescence data found that our algal cells were significantly higher compared to our host tissue data, which helped us identify that ROS occurred from algal cell means. There were no differences found between treatments in season with host tissue, but our algal ROS fluorescence was much higher in the summer collections compared to the winter collections, and no differences were found between treatments. Overall, our linear trend lines show that decreases in ROS fluorescence over time, but show increasing fluorescence in our winter elevated treatment with algal cells. Similarly, with our symbiotic ROS fluorescence data, we found higher outputs from algal cells and no significant differences between treatments and seasons with host tissue. Similar results were found with higher fluorescence in the summer collections compared to the winter collections, and also no differences between treatments. We did find similar linear trends in the summer collections with decreasing fluorescence over time, but found increasing fluorescent trends in our winter ambient treatments. So to summarize our data, we've identified no statistical difference between temperature treatments with our measurements, hypothesizing that Astrangia poculata's thermal limit has not been reached to detect significant differences on overall health. A paper by McGinty et al. 2012 found similar results in our ROS output when exposed to increasing thermal treatments, finding that A. poculata's symbiont, Breviolum physmophyllum, increased antioxidant production to combat ROS. We hypothesized that antioxidant production helped benefit this coral from thermal stress impacts, but we don't have the data to fully support this. In addition, we hypothesized that our maximum quantum yield and photoquantification data had influences from endolithic algae and high nitrate levels to influence these results. Unfortunately, I do not have enough time to explain these in detail, so please feel free to ask any questions related to that in the comments below. Seasonal differences between summer and winter symbiotic fragments were found with our measurements, finding that from the summer collections had higher maximum quantum yield, more symbionts, and had higher ROS outputs compared to winter collections. No differences were found between season and maximum quantum yield and symbiont density with aposymbiotic fragments, but did find similar results with ROS outputs. Astrangia has been shown to go under a quiescent state during the winter months, reducing polyp function, feeding, symbiont density, and other metabolic processes. We hypothesize that this quiescent state influences our results from the winter collections and shows less resilience over time. No significant differences between symbiotic state were found regarding ROS concentrations, except between winter aposymbiotic and symbiotic fragments, where aposymbiotic algal cells had higher ROS output compared to the symbiotic algal cells. We're still determining these results. I want to thank the Global Coral Reef Week for letting me be a part of this and to present my thesis research to the community. I also thank my funding sources, Grand Valley State University and the Michigan Space Grant Consortium for funding this research. Feel free to contact me via Twitter at TylerHarmon9, as well as accessing my website from down below. Once again, I thank GCRW for this opportunity. Please feel free to ask any questions, give feedback, and make any comments, and I look forward to answering them. Thank you.